So today we're going to be looking at the morphology of prokaryotic cells. And we learned in our previous section of notes that the cell shape of bacteria is determined by the cell wall, right? The cell wall is what gives the bacterium its shape. And you probably studied three basic shapes. And that basic shape could have been round, rod, or spiral. So maybe you studied spheres instead of round. And maybe you studied the actual traditional term. Most of the time, we can characterize these bacteria, identify them based upon what shape they have, as well as whether they form clusters or chains. So these characteristics can be used to describe, classify, and identify microorganisms. So why is this important to microbiology? Well, think about it. You're sick, you go to the doctor, they do that fun little swab thing in the back of your throat, and then they'll do a rapid strep test, but they also do another kind of strep test that takes 24 hours. First of all, they grow the bacteria in culture. From there, they put it underneath a microscope. They take a look at it. And then they determine, well, what shape is it? If it's cylindrical, it's not streptococcus. It is not strep throat. So that's one reason why this is important in medical microbiology. So the spherical type bacteria are those round shape. And that word is known as micrococcus. Okay, so you can see right here that this suffix, okay, oops, didn't mean to do that, sorry. That suffix there means spherical. It means the round or spherical shaped bacteria. The cylindrical type is referred to as bacillus right here, okay? And bacillus, being part of the scientific name, usually means that when you go to look at that bacteria underneath the microscope, you're going to see those cylindrical rod-shaped bacteria. Although it's not always used in the scientific name. Escherichia coli, or what we know as E. coli, is also rod-shaped bacteria. So just kind of interesting. So let's take a look at some of the other shapes, okay? The other shapes include what we know as coccobacillus, in which those rod-shaped cells are so short that they're mistaken for round shape. And so when we look up here at this picture, you can see that some of them look like short rods, but when they're turned end to end, they end up looking kind of round, or they're kind of like half oblong, if that makes sense. So that's where they came up with the term coccobacillus. Another shape is vibrio, and vibrio is a short curved rod. So it's not straight underneath the microscope, and each cell is probably going to appear a little bit different there. Now, spirillum are the curved rods that are long enough to form spirals, okay? And so this one over here, this pink example, is a spirillum. You can see that there are some extra structures extending off the ends. What do you think those are based upon our understanding of prokaryotic cell structure? Hopefully, you said flagella. So those are large tufts of flagella at either end of that spirillum. Now, this little guy right here is called a spirochete. Spirochetes typically infect humans. They are long helical cells, and they actually have motility. A lot of times they have little hooks on the end of the cells, which we can't see here in this image, but that enables them to like latch onto certain tissues in the body, which is a little scary. Then there's what's called pleomorphic, and this little guy over here is pleomorphic. He's not round, he's not rod, he's not spiral, okay? He's not any of those basic shapes or a variation of it, so they call that pleomorphic. Now, here's one thing we can tell you for sure. There are groupings in bacteria, and the groupings are determined based upon the way they reproduce. So because most prokaryotes divide by binary fission, that cell splits in half, right? Well, when the cell splits, the cells tend to sometimes adhere to each other after division forming a type of characteristic arrangement. So it depends on which plane the organisms are reproducing. Which direction? Do they split sideways or do they split up and down? Okay? Recognizing the arrangement of some microorganisms can also allow one to make an important identification. For example, Neisseria gonorrhea, the bacteria that causes gonorrhea, can be identified by its diplococcus arrangement. So I want you to predict here. What shape am I going to be looking for gonorrhea? Hopefully you said spherical. And what arrangement? What do you think diplo means, even though we haven't looked at it? Hopefully you said in pairs. All right, so let's take a look at this. One grouping is called strepto because it is in chains, okay? It looks like a string of beads or a string of pearls in a way. And that happens because when we look at it, the cell is dividing along the same plane, and so the cell splits into two. So here's a picture of Neisseria gonorrhea in that diplococcus arrangement. You can see how the cells split, but they're still kind of adhering to each other. Now, sometimes it creates that whole long chain. Then we use that prefix strepto. So streptococcus, the type of bacteria that causes strep throat, makes this long chain-like effect of cells. As more and more cells reproduce, the chain gets longer. Now, there are also what are called clusters or cubes. We really should call them cubes, called sarcina. And sarcina basically divides on all the different planes, creating a cube-like structure, a 3D type structure, which sometimes is hard to distinguish underneath the microscope. So this should actually say two or more planes right there, perpendicular to one another. So they might divide going this way the first time, then the second time they divide this way, then they divide up and down. So that's what creates this grouping of 8 spheres, okay? And hopefully you can see that there's a square, and there's a square, and there's a square. But when we look at them underneath the microscope, isn't that hard to see? You can kind of see that there's a grouping there, and there's a grouping there, and there's a good cube right there. But it's still hard to see. Staphylo is when it forms almost packets. And I actually like to use the term clusters for Staphylo because for me, when I look at this, the resulting cells to me kind of look like a cluster of grapes. And I don't know, maybe it's because this is purple, but we get a single cell that divides on one plane and then it randomly on another plane and then it randomly on another plane and so on and so forth, creating this really random arrangement of cells. So if you were to take right here and just put a little stem up there, wouldn't that kind of look like a cluster of grapes? So when you look at these, this diagram here of Staphylococcus, you can see that they're completely randomly arranged, where when you look back at Sarcina, they're a lot more linear in this picture than they are in this picture.